Thank you very much, ladies and gentlemen. Thanks also to the organizing committee for inviting me to this very interesting congress workshop. Learned already a few things and looking forward to the rest of the program. I will be talking also about the interaction of nutrition with the gut microbiota in early life and focus a little bit on immunity. This my disclosure was already mentioned by the introduction, and what we're looking at, of course, is this very interesting organ that we all need to grow and to sustain, a very complex organ where lots of biological systems come together, lots of neurons, lots of immune cells, a huge surface area where our food is digested, which is not always easy. But now, of course, we also understand that this is also an integrated system with the huge numbers of bacteria that are also there. But okay, today we already have heard quite a bit about neonates, infants, but we all, I think, realize that when a baby is born, the system is not ready, it's not mature. The gut still has to make a few steps to really become the engine of our digestive system. And also this was alluded before. Of course, the planet is a microbial planet, and we evolved much later than the bacteria did. So in order for us to evolve on this planet, indeed, we had to find ways to deal with the microbes that were already there, and indeed build friendships that make us symbiotic organisms that collaborate with the microorganisms. And I think in evolution, we also developed human milk, which is a very special liquid. Human milk does not only feed the infant, it does not only support the growth of the baby, but human milk has also evolved to become a very important microbiome modulator. Human milk drives also this very interesting process, I think, of getting the right microbes into our symbiosis. There's lots of substrates in human milk that are also used by microorganisms, human milk oligosaccharides, for example. There's antibodies in human milk. There is defensins and other antimicrobial properties in human milk. So also human milk is not just feeding the baby, but it also is shaping the microbiome in a very efficient way, depending also of the environment where the mother and the infant are. So human milk is very important also for the gut microbiome. This was already alluded to, so I will not spend much time on this. I think it's now consensus in the scientific world that there is bacteria in human milk, that it's not contamination, but that it's biologically relevant, and that this is also a source of microorganisms for the offspring. And then, of course, human milk does not only contain live bacteria, but also many substrates for bacteria. I mentioned a few, but of course, if you look just at the quantity in human milk, we can clearly see that substrates that can be fermented are a large part also in human milk. So the human milk oligosaccharides, they can make up to 3 to 15 grams per liter in human milk, which is even more than protein in human milk. And most of these substrates are digested, used by microorganisms that are specialized in using these substrates. So human milk clearly directs also the early microbiome. Well, if you look at human milk, which in many cases, of course, is also a basis to feed infants, we know that cow's milk does not contain these complex milk structures. So it's a very different kind of milk that drives also then the microbiology in a different way. And this was also observed for, I don't know, more than 125 years, I think, that the microbiome of human milk-fed infants is different from formula-fed infants. And bifidobacteria is one of these very typical species that you see in healthy breastfed infants, and not so much in infants that just receive a formula based on cow's milk. And as we now all know from genetic analysis, bifidobacteria are really highly specialized in utilizing these very complex sugar structures. They can transport them, degrade them, and use them for growth. So in evolution, again, a mother-infant pair in the environment, of course, gets microbes everywhere, and this is guided. Through inoculation by passing the birth canal. It's guided by the microbes we get from human milk. So the mother's microbiome indeed is a very important source of microorganisms for the offspring. And then, of course, breastfeeding is important. And then certainly also when the weaning foods were introduced, we also exposed infants again to different types of substrates for microorganisms, but maybe also microorganisms. However, if we look in the last 100 years or a bit more, I think a lot has changed in this normal colonization of the gut. We, of course, have antibiotic use. This is very important, it saves lives, but of course, if you use antibiotics in this system that still is establishing, it also has quite an impact on the microbiome. There's studies out there, and I will talk about that later a bit more, showing also that C-section has quite a big impact on the very early microbiome, especially in the first days of life. In some regions of the planet, in some cities, these rates of C-section can be very high, in private hospitals even to 80 or 90% of the infants. Preterm, we know that also preterms, of course, the gut is not ready yet, it's not as mature as it should be, so also in preterms we see quite an aberrant gut microbiota development, and also preterm delivery is actually increasing. So there's quite a few things happening, I think, in the recent history that we may think disturb the normal colonization and also development of the ecosystem in the gut, which is so important because this ecosystem also drives the mucosal immunology in the gut, which is also still immature and goes hand in hand with the microbiome. So also driving the immune system in the right direction, a healthy microbiome is very important. So how can we nurture? Well, these terms, of course, are all known to you. You can feed the microbiome with prebiotics. And actually, we've done quite a lot of work on a mixture of prebiotics, of galacto and fructooligosaccharides. And this indeed is shown to be used as a substrate for beneficial bacteria. And this indeed brings the microbiome of babies much closer to breastfed infants. Also stool patterns, bowel movements, these things are very close to breastfed infants. And if you use these prebiotics in a group of infants that are at risk of allergy, this is a study in Europe, we see that indeed you can stimulate bifidobacteria as a biomarker for, let's say, a healthy breastfed microbiome. But we also see that the cumulative incidence of atopic dermatitis you can indeed significantly impact by using a prebiotic, which is a microbiome modulator. So by influencing the microbiome, you can also influence immune outcomes. Of course, then probiotics, we have also done quite a bit of work on probiotics that might be specifically beneficial and for babies and infants, focus very much on Bifidobacterium breve. And I think we have been studying this bug for more than 10 years because we really need to know what it does it contain any kinds of risks, how does it behave, does it colonize. So a lot of preclinical work has also gone into this strain, and we have a lot of information on this specific strain that was isolated from a healthy breastfed infant. We've done in vitro, in vivo, but also already done some clinical studies, even also in the preterm infants. And we have screened this amongst a whole set of potential interesting probiotic strains. In vitro models, we use mostly mice models, OFA-induced allergy models, and we observed that this strain is specifically suited to influence allergic outcomes. Of course, we can also combine these concepts in something like symbiotics. I think you have two ways of symbiotics. One is the prebiotic has an effect on the gut, and also the probiotic has. But of course, it's also possible that the prebiotic directly can have an impact on the probiotic. So, for example, can be a substrate for the probiotic and even have a synergy. Well, this is caused our interest because we saw in animal models that indeed if you combine a bifidobacterium with a prebiotic, that it's also its substrate of the probiotic, we see an enhanced effect in these animal models. So we've taken that also into a clinical study in C-section born infants. The data are out there. These infants, they have a different microbiome. There's a bit of controversy. If you look too late or not deep enough, you will not see the difference. But especially in the first days of life, the microbiome is clearly different because the microbes are just from the sterile operating environment in the hospital and not from the intimate contact with the mother, of course. Well, we have done a study in Singapore and Thailand with 160 infants where we have indeed done the intervention for a couple of weeks. And as a reference group, we also used vaginally delivered infants as a reference. So we tested a control group, prebiotic group, and a symbiotic group. And indeed, we could confirm in this study that the colonization is delayed in C-section born infants, but that if you use a symbiotic, you already see after 3 or 4 days a significant proportion of bifidobacteria in the gut, and they are active, they produce lactate, acetate, so they're just not passing through because they're eaten, no, they really become active. So these bifidobacteria really find their niche in the gut, and they're very efficient growers under these conditions. So we actually see that with the symbiotics, we kind of correct the early colonization of beneficial bacteria in C-section infants to the level that we also see in a reference group of infants that was born vaginally. So very interestingly, it was not the primary outcome of the study, but we observed that the infants that received the symbiotic also had less allergic symptoms. So they had less eczema, they had less skin disorders in the first weeks of life. And this is generally seen between C-section and vaginally delivered infants. With the symbiotic, we normalized kind of on the frequency of these symptoms, and the symbiotic fed infants are very similar to the healthy vaginally delivered infants. So this, I think, really emphasizes that we should really understand how babies get colonized. We can still learn from nature what's happening, what's the role of breastfeeding. It's going to be very important, because the establishment of the symbiosis drives a healthy microbiome, but also a healthy biological system of us humans as a host. And we, of course, want to build a healthy symbiosis, and for this, the early microbiome development is really important. And of course, we have to find ways maybe to correct for things that go wrong, whether it's C-section or antibiotic use. And I think with nutrition, with probiotics, with prebiotics, maybe also next generation probiotics or fecal transplants, I think if we really understand the mechanisms that are driving the programming of health, I think we can still do a lot more also in the future for the health of babies, but maybe also to have a way to reduce the risk of some of the NCDs, of course, which is also a big burden. For this work, I really need to acknowledge a few people, of course, my colleagues at Wageningen University, but of course mostly also the pediatricians and parents and infants that participated in our study. But I would also like to thank you for your attention and for having me here today.